Verdu.
Hello, everyone. So I will first share my screen. Thank you so much, Mark, for the kind introduction, and thank you.
Oh, sorry. Can you just put it on present view so we can see the whole slide instead? There you go. Perfect.
Okay. Thank you for inviting me. This is really the best audience for me to present, and especially on this topic. So what I would like to do is to briefly discuss the key breakthroughs that have happened in our lab and in our institution with respect to celiac disease, and basically explain how grants that come from outside and more substantial funding really dovetail with donations. And finally, I would like to elaborate on potential future areas of investment that you may want to consider along with Canada Celiac. So let me start by saying that truly building a research program is not a straight path. We think that we get a degree and we get an idea and we start getting results, publishing and getting grants. And certainly this has not been my case. My career has been a little bit of twists and turns, and in letting passion guide my path. And I thought very deeply about this and really what it takes to build a career in a specific area. And the number one thing I believe is having passion for the research area that you investigate, finding the right mentors. But that in red that I put there and I highlighted, obtaining seed funding, I've come to realize that this is fundamental. It is important for allowing you to build resilience, to acquire some importance in the field, building a team of collaborators, and this definitely will ensure more substantial support. And then coming at my stage, it's going full circle and becoming a mentor yourself. So just a few words about my career path. As Mark mentioned, I was MD at the Faculty of Medicine in Buenos Aires in Argentina. I did my residency training, my GI training. I became teaching assistant at the University in Human Physiology. And that picture is the day of my last exam in medicine. I then realized that in order to be a trailblazer and to change things in an area, it was important to acquire research experience. And I went on a grant that I found on my own to a wonderful hospital, University Hospital in Lausanne, that had this combination, which I saw for the first time, where basic immunologists were talking with clinician scientists. And I was very lucky that I met there two people that were then going to become long-term collaborators here in Canada at McMaster University. I even met Dr. Stephen Collins, who was a director of the GI division and also the research intestinal program at McMaster. But life took me somewhere else. I went on after a doctorate and a clinical fellowship to do a PhD in microbiology and immunology. This was the time of Helicobacter pylori infection, a bacterium that caused ulcers. It was a revolutionary time. And so I trained, basically, it was quite a deviation from a clinical career. But this allowed me to then join Dr. Collins' lab in 1998 at McMaster University, and there I am as a postdoc. And so what happened later? Well, in 2006, and this is a long time for a postdoc, I was able to start my first group. And the topic of my research, I decided that it was going to be celiac disease. And I will talk about that in the next slide. Some people looked at me as, you are crazy, you will never get funded by studying celiac disease. This was my first group. It was very small. And just 1 year later, I got the J.C. Campbell Award in celiac disease, where I found people that understood my vision. And so what did I do with this first seed investment? I realized when I came to Canada that there were huge gaps in celiac disease research and also in clinical management. There was poor understanding of the complexity of the impact of this disease here. There were no animal models, no basic research in any university. There was no basic translational research in Canada on celiac disease, and this made me feel really isolated. And this is the reason why mentors, they were well-meaning. They were telling me, I think, Elena, that this is not an area that is conducive to a successful research career. But I persevered. I said, no, this is a gap, and I want to address it. And in fact, I had a vision. I had a vision that was long-term. I thought this is an opportunity to put basic research done in Canada on the scientific world map. It's an opportunity to establish a world-recognized program in translational research. And although these are very long-term objectives, the number one thing that I said, okay, I have to focus and I have to deliver in the short term was to establish and to characterize a relevant mouse model to start to conduct hypothesis-driven research. And I had some ideas. And that's what I did with this J.C. Campbell Award. It gave me that first opportunity to begin to address some tangible gaps and objectives. And so this is what I did. With my initial students here, Heather Galipeau and Justin McCarville, we developed a model in germ-free conditions. These are mice that express one of the genes that are giving the risk for celiac disease, HLA-DQ8. And what we did, we obtained these mice and we derived them without any bacterium or virus. They are basically germ-free. And what this allowed us to do was to reintroduce some microbes. And first we started with a very few microbes, what we call commensals, that are not pathogenic. And we saw that even if we gave gluten to these mice didn't develop any type of disease. Now, when we started introducing microbes that we know cause disease, they are pathogens, we saw that these mice, when exposed to gluten, developed some immune abnormalities that we know are associated with celiac disease, like the increased intraepithelial lymphocytes that are part of the biopsy analysis, gluten-reactive T-cell responses, and antibodies, like, for example, the antigliadin or the TG2 antibodies. So we went further, and what we did was now to take one microbe that we know was associated with the mucosa, the gut lining of patients with celiac disease. And then even if you had few microbes, in the presence of that pathogen, the mice developed some immune abnormalities. And if we took the other group of mice, the ones that had the pathogens, and we gave the pregnant mice antibiotics, and we messed up with the microbial flora even more, then these mice developed even more inflammation when exposed to gluten. So this was the basic studies that this J.C. Campbell Award allowed me to do, and it showed a very important message that gluten pathology in mice that have the celiac risk gene is dependent on the microbes that you have in the lumen. We didn't have any mechanism, but this was sufficient for me to obtain the first Canadian CIHR grant and to elaborate or to come up with a more mechanistic project, and also to be taken seriously in the university. And since now, and until now, I have to say, and I cross my fingers because I have to renew this grant next year, I've had uninterrupted funding to date of CIHRs related to celiac disease programs. So many years later, I had a postdoc, and now I had a system, I had an animal model that we could use to interrogate different questions. And this postdoc that came from Spain, Dr. Alberto Caminero, decided that he wanted to establish a pipeline in my lab to isolate microbes from patients with celiac disease, but from the small intestine, not from the feces. and to study how microbes, for example, specific lactobacillus or model organisms like Pseudomonas that we could find in individuals with celiac disease in the duodenum, whether these microbes could interact with gluten, could use gluten in a way to obtain energy, the same way we do with any nutrient, and then put them in these mice and see what happened. And Alberto actually got a J.C. Campbell Award for all the work that I'm going to describe now. This is a very complex slide, but I give you here the main points. What Alberto found was that gluten can be digested by gut microbes, and specifically by the gut microbes that we can isolate from the small intestine. Now, this digestion is different if you have pathogens or if you have commensals. They degrade it in a different way. And so on the left here, where it says celiac disease, what we found was that those pathogens, although they break down gluten, they don't do it fully, completely, detoxifying the proteins. If we add commensals to that mixture, commensals continue to degrade it, and now gluten peptides are detoxified. And so obviously this had implications in onset of disease, but also in prevention and in therapy, because one could think about exploiting this finding to better degrade gluten through microbes. And this relationship, I mean, what I'm highlighting there, where you have a T cell that is recognizing gluten on an antigen-presenting cell, and then in celiac disease, these cells are going to proliferate and they are going to cause inflammation. We realized that we needed better models to know whether this is only happening in the inside of the tissue or if this is also happening in the epithelial cell. And so that was the new model that we decided to create, a model that only had the gut lining cells, but that expressed those genetic risk genes, and the possibility that that would give us to add microbes, add gluten, and see if we could generate or not an immune response. And this is work very recently published in the top journal of our area, so Gastroenterology, where what Sarah Rahmani, who is a J.C. Campbell Young Investigator Award winner, she used mice, in this case, that express HLA-DQ2. So they are also genetically predisposed. They have the same genes that the majority of celiacs have. And what she did was to take the gut lining and engineer 3D mini guts. These are constructs that will have all the cells of the intestinal lining. And then she can open this, put it in a Petri dish, and now we can obtain cells, T cells that are gluten reactive from the mice that have been immunized to gluten, add them to the system, and we can challenge this with, for example, gluten or with microbes. And what she found was that in doing this, we generate an immune response, meaning that these intestinal lining cells are sufficient to present gluten to these immune cells. And this was really a breakthrough investigation that now is taking us to many other hypotheses and questions. So all this is basic immunology concepts, and I can say that we have our animal models, we have our new models of mini guts, but what's important is to come back to the patient. So we were very good at bringing elements from human disease into models. How do we return to the patient population? And so this was a little bit like the long-term dream that I had at the beginning of my career. And so with Dr. Berchik, who is a gastroenterologist in McMaster, he founded the first adult celiac clinic at McMaster, and this was in 2014. And we realized that without this, we would not be able to do the translation back to humans. And Dr. Burchick had a very talented postdoc and also clinician researcher, Dr. Maria Inés Pinto Sanchez, which you all know, and who recently was awarded the J.C. Campbell Main Award. And so Inés became director in 2018 and really took this clinic to the next level with many achievements that I'm sure she will elaborate on during her talk. And as I was moving up in my career, I was able to incorporate this celiac clinic into a larger initiative that we have at our institute, and that is now funding many of these activities that you have listed there and that Inés will tell you about. So how do we do then the bench to bedside and back? And this was one of the gaps that we also noticed in the microbiome area. Everybody is talking about how gut microbes can influence immune-mediated diseases, like, for example, celiac disease and IBD, but everybody is using stool. And we know that celiac disease affects the upper intestinal tract. So we realized that we had to obtain microbes from these areas. And so we did a systematic study where we obtained biopsies from the first, second, third portion of the duodenum from celiac patients and controls. We sequenced the microbes that are there in the small bowel, and we isolated microbes. And of course, then we fed these to our mice. And one thing that we realized is that, and don't worry about this complex slide, we found that duodenal microbes are very different from those in the stool. So if we are to study the relationship of microbes as a trigger in celiac disease, then we need to go to the duodenal niche. And what we found that these duodenal microbes have less enzymes that degrade gluten. So you can see here the different colors. This dark blue means lower, and all this list are enzymes, microbial enzymes, not our digestive enzymes, that are lower in their capacity to degrade gluten. And when we put those microbes into the mice and we study gluten degradation, we see really that the mice that are colonized with those microbes that lack that enzymatic activity have lower capacity to degrade gluten. And so this really completed the full circle into mouse to human and human to mouse. So this takes me to another important breakthrough in microbial metabolism. We are in general interested in how our gut microbes contribute to prevent disease or to make it worse. And actually, they do the two things. And one of the metabolites that we are very interested in is this essential amino acid that is called tryptophan, and that is present in many different foods, but it regulates many functions. It regulates mood, and we heard from the previous work, the mental fog, and so on and so forth, anxiety, depression associated with celiac disease. But tryptophan metabolites also regulate inflammation and other gut functions, and we need to obtain it through our diet. And we also know that this metabolic activity of the microbes in some immune-mediated diseases like IBD is impacted, it's altered. So we decided to study whether this was also affected in patients with active celiac disease, because we knew they had different microbes, they metabolize gluten differently. Could they also be metabolizing tryptophan in a different way? And indeed, that's what we found. The metabolism of this amino acid that comes from food sources is impaired in celiac disease. And what happens is that because this amino acid is not being chopped up by microbes as efficiently as it should be done, we get less metabolites. Metabolites are the products of the digestion of that amino acid, and that can promote inflammation. And what was interesting is that even in patients with gluten-free diet, the pathway was better, but it was not completely restored. And so how can we impact this? And this is an example that I'm giving you of a basic discovery to taken to the clinical scenario, because we have now the pipeline to do that. And Gaston Reda decided first to say, well, is it safe, and can we actually improve or have a measurable effect in the proximal small intestine if we give this amino acid to healthy individuals? And so he did this study where some individuals got placebo, others got tryptophan. And the idea was to deliver this amino acid not in foods, but in pills that would be dissolved right there in the small intestine, with the idea to see whether microbes in the small intestine can produce these metabolites. We can measure that, and if this was safe. And indeed, his study, which was published very recently, showed that individuals getting this treatment did not get any gastrointestinal symptoms. It actually stimulated this metabolite production. We could measure that. And this implies that it could be a therapy for patients with non-responsive celiac disease. And so that's what we are doing now. And this is UK in my lab. She's a PhD student, and she's been conducting this study where we basically have the same plan to give these pills of tryptophan that dissolved in the proximal small intestine, but this time we are doing this in patients with celiac disease that are on a gluten-free diet, but that they have borderline antibody production or are still symptomatic. And our primary objective is whether we can improve those symptoms, but we have objective measurements, secondary objective measurements, which is this increase in the metabolites produced by the indole, because that would be telling us that the tryptophan is being metabolized by the gut microbes. And so we are currently recruiting this for this study. And so I'm going to give you some interim analysis. This has not been published, so please do not post in any social media, just this slide. But we've done an interim analysis, and it's very promising. We see that we are halfway in the study, and the patients that mainly receive tryptophan have a higher percentage of being asymptomatic if they were receiving tryptophan as compared to placebo. What is very interesting is that this is also the same trend when we measure gastrointestinal symptom rate scale, and especially the quality of life has improved 58% of the subjects on tryptophan compared to none receiving placebo. So we are very, very excited. We still have to continue recruiting in this study, but UK was awarded the J.C. Campbell Research Main Award, and she will be completing this study thanks to this grant. So let me now go to the final part of the talk. Donations and the J.C. Campbell have been really impactful. And I tried to think, okay, what are the main areas where the impact has been significant? And I think it has been in terms of community building, because getting these grants, these awards, truly draws attention to celiac disease nationally. And I've seen throughout the years how many other universities have become interested around Canada in celiac disease research and clinical care. It gives us national recognition, and that significantly increases the competitiveness of these awardees. And you saw the example of Alberto Caminero. He's now an investigator in his own right with his own research program and a very close collaborator. He's an associate professor at McMaster University. And of course, it advances celiac disease research because this is seed money, but I tried to show you how seed money can help us obtain further funding. Now, research is expensive, and we can talk that if we have time for questions. So I really have to thank the celiac community in Canada, and especially CCA, for supporting this vision. These are all listed here from 2007, the very first J.C. Campbell Research Award to myself, how then so many trainees in our group have received this award, and even the young investigators. And I could tell you exactly where all these students are, and you would be very proud to know where they are, except for Mark. Mark is very recent, and he's still doing his PhD. So highlighting again how, yes, research is very expensive, and you would say, well, a 25,000 or a 5,000 award is not enough. It can make a huge difference, that seed funding, because it will link through all the points that I mentioned about ensuring more substantial funding in the future. So are there areas of support for direct transformational impact that we could work with? So yes, celiac, research is really very expensive at the basic and at the clinical level. And yeah, we need more support for basic discovery. National agencies have their own priorities, and celiac disease, unfortunately, is not one. So what can we do about this? Well, I think that we could try to lobby for targeted fundraising. So donors that care about this, and lobby CIHR and government for celiac disease initiatives. These do not exist currently, and such as, diabetes has, cardiovascular disease, cancer, and many other areas, but we don't have celiac initiatives in CIHR. We could leverage also academic center investments. So instead of just giving one grant, one could think about match investment and ongoing successful programs that have match, that have funding, and you could match to accelerate that pace of discovery. And I'm giving you some big picture ideas. It doesn't have to be this, but one could think about establishing professorships in celiac disease. These things exist in IBD, for example, and really that commits the career of a person towards investigating in that area. Or you could simply try to see if you could be a partner for a chair in celiac disease with someone else, or explore collaborations with similar-minded foundations. And I just bring the idea of the Western Foundation because they are interested in nutrition and food and in how it modifies our microbes. And so maybe there could be opportunities to discuss with them and have priority funding for celiac disease. So I just want to end with saying this is who we are today. We are an interdisciplinary, multidisciplinary institute at McMaster University. I have been honored with taking over the direction of the institute in July of this year from Dr. Collins, my longtime mentor. And this goes back to the idea of having the right mentors, but as well becoming one at the end of the path. And so I want to leave you with what the current initiatives at the Farncombe Institute are. We are very strong at lab research, very strong at clinical research, but we did realize one year ago that we had to make that move stronger. So from lab to clinical and from clinical to patient care. And we proposed to have new initiatives, and we have 2 new initiatives in the Institute that are funded by our main philanthropic donors, the Nutrition Initiative and the Microbial Therapeutics Facility. And this, I think, will integrate well with continuing really that dream that we have of impacting patient care and community health in general. And so I just want to leave you with some photographs of trainees that went through my lab. All of these obtained research funding from CCA, and that are now have graduated, left, some have stayed, like Dr. Caminero, but they all have very successful careers thanks to your investments and your donations. Thank you very much.
Okay, thank you, Dr. Verdu. Just going to give away a couple of prizes before we get to Q&A from a cut.